Chapter number 12 is on protein sorting and transport. There are 4 parts of the chapter, and we'll try to cover most of them, except a few pages that you will see actually on the next slide. So these are the 4 parts of the chapter. We'll talk about the endoplasmic reticulum, Golgi Apparatus, and how actually transport takes place inside the cell. And the last part of the chapter talks about how proteins are degraded. A lot of time proteins that are produced are not the correct type of proteins, so they are recycled. First, they are broken down into individual amino acids or polypeptides, and then amino acids, and then they are recycled. So that's what we will talk about in the last part of the chapter. Here are the pages that we will not be you are not responsible for preparing. I'm not going to be talking much about from those pages, and you are also not responsible to prepare those for your next exam. There are many proteins that are produced in prokaryotes as well as in case of eukaryotes. In case of prokaryotes, proteins are produced in the cytoplasm, but not all proteins are needed inside the cytoplasm. They are designed to go inside different organelles. They are targeted to go inside different organelles from cytoplasm. Even those proteins that are meant to go inside nucleus are produced inside the cytoplasm. Since nucleus does not have its own machinery of production of the protein, so they're all produced in the cytoplasm. Same thing is true in case of a Golgi apparatus or endoplasmic reticulum or other types of organelles. And all these proteins that are produced in the cytoplasm follow a very specific pathway. And most of the time proteins are enclosed. They are not really naked proteins. They actually are enclosed in tiny vesicles, and then they are transported from one place to the next place. So endoplasmic reticulum is one of the organelles. This is the largest organelle. It is membrane-bound structure, very extensive inside the cytoplasm. It almost covers the entire cytoplasm. It is connected to the nuclear membrane, or you can say it is continuous with the nuclear membrane. Nucleus has 2 membranes, inner and outer membrane is actually endoplasmic reticulum. And so because of the role endoplasmic reticulum plays, it is the largest organelle inside the eukaryotic cells. There are 2 types of endoplasmic reticulum, or you can call it endoplasmic reticula. And one is called rough because of the presence of ribosomes attached to it. Another one is smooth because of the absence of ribosomes. Both of them play very different roles. The role of smooth is not related to proteins. It is mostly related to lipids. And the role of rough is in the production of the proteins because of the attachment of ribosomes to endoplasmic reticulum. So that's the main role of both of those. One is related to proteins, which is rough, and other one is related to lipids, which is the smooth one. So protein production on endoplasmic reticulum was the pathway was discovered by Polade and his colleagues back in 1960s. They were working on the pancreatic acinar cells, and acinar cells, if you know, are the cell that secretes the digestive enzyme, which is the most kind of a broader and most effective type of digestive juice that is produced by the pancreas. So they were trying to follow, since those cells, acinar cells, are directional cells, they are polar cells, they are attached to the membrane, but then they secrete their proteins and enzymes only in one direction. So Polade and his colleagues they were trying to find out if they can trace the movement of the proteins, because they know these cells are directional cells, these cells are polar cells, so they are attached and they release their produce only in a specific way. So they thought they can actually uh, use this as a way of finding the movement of proteins that are produced inside these cells. So what they did was they they used some radio labeling to find out the movement of the proteins. The experiment that they did is called pulse chase experiment, and chase experiment is based on feeding the cells with radioisotopes of amino acids and then following the proteins that are produced inside the Golgi, inside the endoplasmic reticulum, and see where they go. So initially they would feed the cells with radio-labeled amino acids, and then they will stop doing that. Then they will let the cells produce the regular types of proteins without radioactivity on them, and then at different time points they will follow where those proteins are moving. That way they were able to find out the secretory pathway of the cells. And that pathway is based on this pulse chase experiment that they did on acinar cells. So now we can actually see that pathway. The pathway is, initially they found out within few minutes after they fed the cells or after they used radioisotopes on amino acids or amino acid labeled with those radioisotopes, they found that most of the radioactivity was concentrated very close to the nucleus, where endoplasmic reticulum, the rough type, is mostly concentrated. And then after some period of time, within few minutes or so, they found out that radioactivity was mostly concentrated away from the nucleus. So this is where all the network of rough endoplasmic reticulum is located. So that happened within few minutes after giving cells those isotopes, radioactive isotopes, and then few minutes after that, not actually several minutes after that, they were able to see that radioactivity was leaving the cells. So this actually shows the the polarity of the cells. Cells are attached to the bottom. So here the cells are attached. So there is only one way to secrete, to release their proteins, which is towards this side. So this side is where it is connected to the ducts, and ducts carry that enzymes from the pancreas and dump it into the first part of small intestine. So in a couple of minutes, a couple of hours after they found the radioisotopes or radioactivity in the Golgi apparatus, they found out that radioactivity was leaving the cells. So they designed, or they came up with this idea that the pathway, the secretory pathway, is that it is produced, proteins are produced in rough endoplasmic reticulum. From there they move to Golgi apparatus, and from Golgi apparatus they are enclosed in tiny vesicles through which they leave the cells, and they are dumped in the ducts, which is carried to the other parts of the body from there. And so this was true for those proteins which are considered as secretory proteins. But then in a study that was done after this initial experiment established that it was also true for the non-secreted proteins, which are not released. They are not released from the cells. Insulin, for example, is released, and these digestive enzymes are released from the cells. So are many other hormones that are released from the cell. They follow the same pathway. So the pathway is a rough endoplasmic reticulum to Golgi, and from Golgi to secretory vesicles, and then outside of the cell. So sorting takes place at different levels. Some proteins are synthesized on free ribosomes. They generally stay in the cytosol, or they can be transported to the nucleus or to some other organelles. Proteins that are synthesized on the membrane-bound ribosomes are translocated directly into the endoplasmic reticulum. So their pathways, they first go inside endoplasmic reticulum, because ribosomes that are attached to endoplasmic reticulum is where they are produced. So the easiest pathway for them is to go inside endoplasmic reticulum in the lumen, and then gets modified from there, and then they follow the rest of the pathway. So here there are two different types of ribosomes, as we are familiar with that. One type of ribosomes are the free ones. The products that are produced on the free ribosomes are designed to go inside nucleus, mitochondria, chloroplast, or peroxisomes. And the products of those of the ribosomes that are attached to endoplasmic reticulum, they go inside Golgi, and then they are designed for nuclear membrane or plasma membrane or endosomes, lysosomes, peroxisome membranes, mostly the membranes. So most of those are membranes, like these are membranes, and endosomes. Endosomes, we will learn later on, they actually give rise to lysosomes. Otherwise, plasma membrane or nuclear membrane or peroxisome membranes are just the membranes through which endoplasmic bound ribosomes produce proteins, and then they directly get embedded in the membrane, and then they become part of the membranes. And that does not happen. The membrane-bound proteins are not produced on the free ribosomes, because free ribosomes do not have this double layer. Actually, it will become more clear later on what is the significance of this membrane, because proteins that are produced, they get embedded directly here, and then these membranes become part of the vesicles that carry them, and those vesicles become fused with those membranes, and that's how those proteins become part of those membranes. Targeting those proteins is done with the help of signal sequence, a small sequence of amino acids which is present mostly on the N-terminus of the proteins. So N-terminus of the proteins indicate where those proteins are designed for. Proteins go inside mitochondria or nucleus. They have different types of sequences, as if they have different types of zip codes. So zip codes, or those signal sequences, actually target them to specific types of organelles. So this type of information was obtained by preparing those in vitro type of preparations of the endoplasmic reticulum. So chapter number 2, we learned how this cellular fractionation works. When we fragment cells, they're broken down into individual organelles, and at the end what's left are those tiny microsomes, which are the products, or which are the fragmented products of endoplasmic reticulum. Since there are two types of endoplasmic reticulum, they can be separated from each other. They're both very small in size, but the one which have ribosomes on them are generally heavier than the other ones. So those are the ones that are collected towards the bottom of the tube, and the smooth ones are a little bit above that. But both of those can provide some, and you can actually separate them from each other and use the one that have that are rough endoplasmic reticulum for translation of the proteins. And this type of experiment was done by Blobel and Sabatini in 75, that they found out that secretory proteins on the free ribosomes retained the signal sequence and were slightly larger. So, they compare the size of the proteins that were produced by the ribosomes, which were free floating inside the cell extract, as compared to the one which were produced on those tiny microsomes that have in which the ribosomes were attached to the endoplasmic reticulum, and the size of those proteins was a little bit smaller because of the truncation that happened or processing that happened on those proteins. So something like this that can be seen. So if those are free ribosomes, there is no mechanism of breaking the signal sequence. Signal sequence, which takes the proteins to their destination, like in this case here. And in case of lack of membrane-bound ribosomes, those small sequences are not removed. That's why the size of those proteins is a little bit longer as compared to those proteins that are produced in membrane-bound microsomes. Because microsomes have this mechanism of cleaving that small piece of signal sequence with the help of an enzyme, and then after that happens, the proteins become localized. Like in this case here, in this experimental situation, this protein will not leave this microsome, this small vesicle, because a signal sequence has been lost. Signal sequence is generally very small. It's about 15 to 40 amino acids altogether, and they usually have a long stretch of 17 to 12 hydrophobic sequences. And generally, it is located towards the end terminus of the protein. So most of these sequences that we see here in the middle, those are the ones that are hydrophobic in nature. So as the proteins or polypeptides emerge from the ribosome, signal sequences are bound by those receptor proteins called signal recognition particles, SRP, and those are the ones that attach to those polypeptides that are newly synthesized, and then they are the ones that take them based on the signal sequence to their destination. And these recognition particles or proteins, they consist of 6 polypeptides and small cytoplasmic RNA molecules. So those are ribonucleic proteins. Those are RNPs. They have both proteins and RNA molecules in them. So in the mechanism actually shown in the next slide, so that process is called as co-translational pathway. So as they are synthesized, as they are being translated, they are being carried to their final destination at the same time. So SRP carries them, those protein particles, recognition particles, carry them. They go and attach to the membrane. Usually there is a small hole, small channel in the membrane called translocon, like in case of endoplasmic reticulum. Then the signal sequence, which is on the end terminus of the polypeptide that is being transported inside endoplasmic reticulum, is inserted through that translocon protein, small hole, small channel, and then as it enters the translocon, it is cleaved. So as a small part of that goes through that translocon, it is cleaved with the help of an enzyme attached to the membrane called peptidase, signal peptidase, which is specific for removal of the signal sequence, and that is located inside the lumen of endoplasmic reticulum. And then the rest of the polypeptide is inserted inside, and then after that, once it's inside endoplasmic reticulum, inside the lumen, then the protein cannot leave. Now it has lost its zip code, and any letter that does not have a zip code really goes nowhere. If you have a letter that has a zip code on it, no address, nothing else on it, you will find that letter in that zip code. For example, if somebody sends a letter, from puts a stamp on it, puts a zip code 28301. So that letter will be here in Fayetteville, and it will not go anywhere from there, but it will be here in the Fayetteville area, because it has that zip code. So those proteins that have that signal sequence on it, they have that zip code, they actually find themselves inside those organelles, and once the signal is removed, then they cannot move anywhere else from there. This is the whole mechanism. So in this case here, ribosomes are translating a small part of the protein that we can see coming out of the ribosome here. So that's, and that the first part of that protein is the signal sequence, and then that small signal sequence is recognized by SRP. The particle binds to it and then brings it and attaches it to this receptor. So that receptor is present on the surface of endoplasmic reticulum, and then after the attachment, the small signal sequence goes inside the translocon. Initially, translocon is closed, and when the attachment of this protein to the receptor takes place, this gate opens, and through that gate, the proteins come, and the polypeptide starts to come through. And on the other side of this translocon protein is a small sequence of a small peptidase, a small enzyme attached to that. As the signal sequence comes through, this peptidase recognizes it, removes it, and then rest of the polypeptide comes through. And once it is there, then it gets trapped. This is where it is destined to come. This is where it was targeted to come. So proteins that are synthesized, some of those are meant to go to become part of the membranes, because as you know, most of the membranes consist of two things: phospholipid molecules and proteins. So those proteins are produced in those ribosomes which are attached to endoplasmic reticulum. They become part of the membrane directly from there, and then they are carried to those organelles as vesicles that have those proteins already embedded in them. And another thing which is important to know is that the transmembrane domain of the protein consists mostly of the alpha helical regions, which consist of those amino acids which are hydrophobic, because the interior part of the phospholipid molecule is hydrophobic. So they become part of the interior part of the protein. And then the orientation wise, it could be both orientation. C could be on the outside of the membrane, or C terminus could be inside of the membrane. Either way, different proteins are different in that regard. Their orientation is different. Some have N terminus outside, and some have their carboxyl terminus in the outside. So for example, here there are different proteins shown here. One has the C domain on the outside, other one actually has its N domain on the outside. Some of the proteins, they go through transmembrane fashion only once. So this is their helix, alpha helix, embedded in the hydrophobic part of the membrane. In other case, another protein that is shown here, this protein has kind of transversed several different times. This is one here, and then goes, okay, if you go from N terminus towards C terminus, that's the direction of the first helix, and the direction of the second helix is opposite, and then the third one is opposite, and then the fourth one is opposite. So they're just going kind of anti-parallel way, because it's going through the membrane multiple times. So it is going, the alpha helices are anti-parallel to each other. So what's inside of the endoplasmic reticulum is the same thing as the exterior of the cell. Because if you think a vesicle which is produced through the secretory pathway is carrying a protein on it, becomes enclosed in a tiny vesicle, and then that gets fused with the outside membrane, which is the plasma membrane or cell membrane. So what is enclosed inside that vesicle is the outside of the cell. How is that possible? Let's take a look at actually some of the vesicle that is being produced here. In this case here, this vesicle, which is shown here, is carrying a protein. This blue thing is a protein which is produced in the same fashion as we saw in the previous slide. So that protein in this case is embedded in the membrane. That membrane is enclosed coming from endoplasmic reticulum. It gets enclosed in a tiny vesicle, and you see the orientation of the protein. Okay, so orientation of the protein is on the inside is this smaller part of the protein. So this is kind of a knob-like protein that has a smaller piece in the inside of the vesicle. The vesicle becomes part of the after endoplasmic reticulum. It becomes part of this Golgi apparatus. And then from here, it is enclosed again in vesicle. So, when proteins are transported, they are enclosed in endoplasmic reticulum in a vesicle, and then they become fused with the Golgi apparatus, and then they leave Golgi apparatus is tiny vesicles again, and then the final destination in this case happens to be the membrane, the plasma membrane. So this tiny vesicle becomes fused with the membrane, and when it becomes fused with the membrane, it becomes fused in such a way that this part goes to the outside. So the interior part, let me actually take a different color if I could. So this tiny internal part of the protein, which was inside, now has become the outside of the membrane that you can see here. That's what it means, the orientation of these proteins. What is inside of the vesicle is the outside of wherever they dock to. If they dock to, or if they fuse to the external membrane, then whatever is on the inside of the vesicle becomes the outside of those organelles, or in this case here, in this example, is the plasma membrane. There are two types of polypeptides. Polypeptides that have a signal sequence on their N-terminus, and some polypeptides do not have that signal. So the one that have the signal sequence on them, they are recognized by the receptors. Receptors bring them to the translocan, and once they start inserting the polypeptide through the translocan, then they are terminated with the help of that peptidase, which is present on the lumen side. This is something similar that we have seen in the previous slides also. But then there is an internal sequence in those polypeptides also, because these proteins are designed to be embedded in the membranes. So that internal sequence is recognized, and then instead of kind of inserting them towards the lumen of endoplasmic reticulum, it is kind of retained and pushed towards the membrane. Instead of kind of pushing them through this translocan, all of the protein, once that signal sequence is there, internal signal sequence is there, then instead of kind of inserting them into the lumen, they are pushed to the side, and they become part of the membrane. And orientation again could be either way. It could be N-terminus on the outside, or C-terminus on the outside, and the opposite terminus could be on the inside. Then there are some other proteins that do not have that signal sequence on them. Those signal sequences are recognized by the receptors, which are present on the surface of the membranes. And then as they are pushed through the translocan, the internal sequence is revealed, and that internal sequence pushes those proteins to the side, and they do not get inserted into the lumen. Because once they're in the lumen, then they're enclosed in the vesicles, and then they're transported to the Golgi apparatus. But these proteins are meant to become part of the membrane. They become embedded in the membrane, so they are pushed towards the membrane of endoplasmic reticulum. And when the vesicles are made from the membrane, then those are enclosed in those vesicles, and then those vesicles are carried to the final organelles where they become embedded in those organelles. So there are different arrangements how they become embedded. Some proteins have only one transmembrane domain, as we saw before. Other proteins have multiple domains. They actually cross the membrane multiple times. Some of them have carboxy terminus on the outside, some have N-terminus on the outside. So they're all different proteins, and their mechanisms are slightly different from each other as well. So for example, this one is showing the orientation, opposite orientation of two different proteins here, one that has N-terminus on the outside and the other one that has C-terminus on the outside. So though the way they are kind of grabbed and are made part of the membrane is slightly different from each other. So once proteins are produced, then the next thing that happens to the proteins is that they are folded in a proper way. Proteins are originally, as we have learned before, proteins are protected from folding initially with the help of attachment of chaperone proteins, and then later on those chaperone proteins detach from the polypeptide and allow those proteins to fold in three-dimensional way so that those proteins can become functional proteins. So chaperone protein's role is to allow the formation of three-dimensional structure of the protein. So here's one protein shown here. This is a heat shock protein. Chaperone proteins called BIP proteins. So it binds to the proteins that are being inserted in the lumen through the translocon in the lumen part of endoplasmic reticulum. So BIP protein binds to it and prevents it from forming the three-dimensional structure. After the entire length of the polypeptide is inserted through the translocon, then these chaperone proteins detach from the polypeptide, and based on the sequence, based on the primary sequence of the amino acids in polypeptide, proteins fold to make a three-dimensional structure. And then after that, the next stage is to export those proteins. So those proteins which are meant to be exported, so now actually we have skipped a couple of pages here. We jump to page number 402. So that shows, how the export of proteins and lipids take place from endoplasmic reticulum, and those things are exported by enclosing them in tiny vesicles. Almost there is no transport of things that take place inside the cell which does not follow the vesicles. Things are enclosed in the vesicles, and then they are taken to their final destination from there. So from endoplasmic reticulum, the vesicles that are formed, they bud out from the surface of endoplasmic reticulum, and then they exit through a site which is called ERES, which is endoplasmic reticulum exit site. So ERES. And then they actually become part of a temporary type of an intermediate compartment, which is called ergic. This is endoplasmic reticulum, Golgi intermediate compartment. So kind of a mouthful, but that's kind of a transitionary path which is present between endoplasmic reticulum and Golgi apparatus. So there is some recycling that is done in ergic. That's why ergic is important. So here, the whole pathway is shown on this slide here. Whatever is being produced, in this case here, in endoplasmic reticulum, is leaving endoplasmic reticulum in the form of these tiny vesicles that are budding out of the surface. They're coming out of this exit site on endoplasmic reticulum, which is located closer to Golgi. So this is Golgi here. And this is the intermediate phase in between these two, is called ergic. So vesicles that bud out from ERES, they fuse with ergic, And ergic is a transitionary phase in which the recycling takes place. Anything which is useful for endoplasmic reticulum is cycled back towards endoplasmic reticulum. Otherwise, from ergic, they are enclosed in tiny vesicles again. So, and then they fuse. Those vesicles fuse with Golgi apparatus. A lot of times, since the vesicle that bud out from endoplasmic reticulum carry those proteins that are shown here, which are part of endoplasmic reticulum. So those proteins are recycled by making tiny vesicles and fusing them back to endoplasmic reticulum. So those proteins are moved back where they belong to, because the purpose was not to lose those proteins. The purpose was to transport things inside the vesicles, and the vesicles, once they were formed, they contain those proteins, and those proteins are moved back into endoplasmic reticulum, because that's not the product that needs to be released from endoplasmic reticulum. What is released is what's inside those vesicles. So that's how kind of a recycling is taking place on them. And then how do they know what type of proteins are part of the endoplasmic reticulum? That is based on this small sequence called KDEL. Those are the 4 amino acids in those proteins, and that is lysine, aspartic acid, glutamic acid, and leucine. Those 4 nucleotides, when they are present, that means those proteins are the receptors present on the surface of endoplasmic reticulum. So cells recognize them and retrieve them back to endoplasmic reticulum, where those proteins become part of the membrane again. So all proteins and lipids are transported the same way. They leave endoplasmic reticulum in tiny vesicles. They fuse with ergic, and from ergic they become part of Golgi apparatus. Anything which is meant to be moved back to endoplasmic reticulum is enclosed in tiny vesicles and fused back on endoplasmic reticulum. So this slide just shows that sequence of KDEL, which is lysine, aspartic acid, glutamic acid, and leucine. Showing the same thing again. This is how things are kind of moved back. So I had duplicated this slide here, the same slide that we just seen a couple of 2 slides ago. Okay, now the Golgi apparatus, the second phase before the proteins are secreted out of the cell is the Golgi apparatus. Golgi apparatus receives all those vesicles coming from endoplasmic reticulum, and then Golgi apparatus kind of sorts them, modifies them, enclose them in vesicles, and send them to their final destination. Some proteins are designed to go outside the cell. Those are the secretory proteins. Other proteins are designed for internal destinations. Golgi apparatus, so that's one important role Golgi apparatus plays, which is modification of proteins and kind of shipping them to their final destination. Another important role of Golgi apparatus is this is the site of the production of glycolipids and sphingomyelin. Both of those that are part of the cell membranes. And also polysaccharides are produced here, which are designed for the cell wall in case of plant cells. In the structure of Golgi apparatus is more like, kind of stacks of pancakes, which are put right on top of each other. And all of those pancakes are kind of separate from each other, but once they're placed in a stack, it looks like as if there's a part of the same body. And Golgi apparatus also has a polarity. It has kind of an entrance and it has an exit. Both of those are labeled as cis site and the trans site or trans phase of the Golgi apparatus. So this here actually we do see the 2 sides. So it has basically 4 different locations or 4 different sites. Cis site. Cis site is the one which is the nearest to the endoplasmic reticulum. So that is basically the entrance, the entry point for the, or the fusion point for the vesicles. And then there is a medial and trans site. And then the last site is the trans-Golgi network. So this is where the vesicles that came through the cis site, and they went through, going through different layers through medial and trans, and eventually they were enclosed again in tiny vesicles to the other side. So they received from one side, which is this side here, and these were kind of remade on the other side. But now what has happened in this process is a lot of modification took place on those proteins which were brought in from endoplasmic reticulum. Each layer inside Golgi apparatus has a different modification mechanisms in it. Proteins go through many different modifications, and different locations carry out those modifications on the proteins. For compartments, so there are two that are kind of fused together, shown here as the medial and trans compartments. And then the cis one is the entrance point, and then the trans-Golgi network is the exit site of the Golgi. So proteins that are produced in endoplasmic reticulum, they're enclosed in vesicles, and then those vesicles arrive at Golgi apparatus. Golgi apparatus has modified those proteins and is ready to be transporting them out of the Golgi apparatus. There are 3 routes that are followed. One route is proteins are directly released to the plasma membrane. So those are the things that are released directly. They are not contained inside the cell. The second one is through recycling endosomes. This is a kind of a mechanism in which vesicles fuse with a structure called endosomes, and then recycling takes place. Proteins that are needed to stay inside the cell, they are moved back to their own destinations, like in case of Golgi apparatus. Some proteins that are in those vesicles are Golgi resident proteins. So those are recycled, enclosed in tiny vesicles, and those vesicles fuse back with Golgi apparatus. The third types of proteins are the proteins that are secreted through a regulated pathway, meaning that those proteins are produced to be released, but they are not released right away. Like insulin. Insulin is produced and enclosed in tiny vesicles and released outside the body when glucose is present in the bloodstream. Or in case of immune cells, those pharmacological compounds are produced and kept inside the white blood cells and released only in response to allergens, for example, or other types of threats that are faced by the immune cells. So those 3 pathways are shown here. The pathway of the secretory proteins. One is that becomes part of the membrane right away. This is that pathway. So produced, enclosed in vesicles, and fused with the outer membrane, the products are released to the outside. The second one is a regulated pathway in which vesicles are produced and are kept inside the cell and released only when they are needed to the outside. And the third pathway that is shown is by fusing to the endosomes. Endosomes, or recycling endosomes, there are 2 types of those are shown here. One that results in the production of the lysosomes, other is the recycling endosomes. So these are 2 different types of endosomes that are produced. So here the purpose is to use it to recycle some of those proteins that are released from Golgi, and usually they are moved back towards the Golgi as well. Once they are recognized here on the endosome, then they are enclosed in the vesicles and fused back. The same mechanism is used here to differentiate those proteins which are kind of resident proteins of endoplasmic reticulum based on their amino acid sequence. So if those amino acid sequences are there, it is understood that those proteins belong to Golgi apparatus. So those proteins are enclosed in vesicles and fused back to Golgi apparatus. So this slide is talking about regulated pathway, like followed in case of hormones, insulin, for example, or other types of hormones, or neurotransmitters. Neurotransmitters are also not released simultaneously. They are produced by the neurons, and then they are enclosed in vesicles and released only at the time when they are needed.